Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome everyone back to the Genetics Podcast. I'm really excited to be here today with Dr. Hilary Martin, who's a group leader in human genetics at the Wellcome Sanger I. She's focused on analyzing large-scale genome sequencing and genotype data, in particular neurodevelopmental disorders, but also in populations where consanguinity is common. We're going to dive into some of Hilary's recent work. This is my first recorded conversation with Hilary, but far from my first conversation. When I was a PhD student in Matt Hurl's group, Hilary was one of the postdocs, but shortly after I left, she was promoted into a group leader position, which she's been doing for the last 6 years or so. So I've had the pleasure of being directly in the same room with Hilary for a very long time and learned a lot from her. So I'm looking forward to this conversation. It's maybe 6 years too late, but thanks.
Well, we missed you at Sanger, Patrick. It was a golden era when you were there.
They're great. There've been many golden eras at Sanger, but I appreciate it. I enjoyed my time there and I learned a ton from everybody, but in particular, you always had an incredible amount of energy. One of the things that is hard, I think, for some people transitioning from postdoc to group leader is coming up with their own ideas. But I suspect that probably wasn't much of a challenge for you because you were always full of creative ideas. Was that the case at the time?
Well, you hide it. I must hide it well then, because to be honest, I do find it quite hard coming up with feasible big ideas. I mean, it's not difficult to come up with sort of incremental ideas. What would I do next given what I've just done? But coming up with a big long-term research program that's sufficiently ambitious and feasible is not easy, actually. And I really felt like I was kind of winging it back in, was it 2018 when I applied for this position? But anyway, I seemed to fool people, and here I am. And in the last year, as I was saying just before, things have finally started to sort of come to fruition, and I'm feeling more confident now, I guess.
Yeah, that's great. I mean, maybe there's a little bit of imposter syndrome there, but I from my perspective, you were always ready and have done some amazing work. So clearly the proof is in the pudding. Could you tell people a little bit about what your group focuses on, a little bit more detail for people who aren't familiar with your past work?
Sure. Well, we have two strands, really. Half the group works on neurodevelopmental conditions and traits with a focus mostly really on understanding the genetic architecture broadly. And the other half works on mostly complex diseases, but particularly in populations with high rates of consanguinity. So we have a big focus on this Genes and Health study, which is based at Queen Mary University in London. So at a high level, that's what we do. And I'm sure we'll go into some more details.
Yeah, let's start on the first strand. You wrote a really interesting paper recently that was published in Nature that dove into rare and common variants in their role in neurodevelopmental disorders. I think that paper is probably a good lens to maybe catch people up on where that field is as a whole, especially in the interplay between common and rare genetic variants. I wonder if you could just start with there's an initial strand of this, I guess, which is that most people think, ultra rare and damaging variants are the primary cause of developmental disorders, which is the case, but that's often caused common polygenic risk to be overlooked. And your recent paper did a really good job, I think, at unpicking what the role of common variants are in this. So maybe you could start with what was known before the paper and then talk a little bit through what you found. And there's quite a bit in the paper, so I think we'll spend some time on the three different findings.
So what was already known about the genetics of neurodevelopmental disorders, I guess I should just give that a bit briefly, since this is a broad audience. So we knew that there was a very large Mendelian component. So work from your PhD lab, Matt Hurl's group, has estimated that probably about 40% of cases with developmental disorders can be attributed to de novo mutations in coding regions that we assume are kind of monogenic causes. And as your PhD work showed, probably de novo mutations in highly conserved regulatory elements explain a pretty small fraction. I think it was sort of on the order of 1%. We also know that in populations with low levels of consanguinity, monogenic recessive causes explain maybe 3% of cases. That's much higher in populations with high levels of consanguinity. And we knew that X-linked coding variants anyway explain about 7%. So in total, maybe half of cases with these conditions can be attributed to monogenic causes in the coding regions. But in about 8 to 10 years ago, I guess Jeff Barrett, who was my postdoc mentor, said, well, maybe common variants also contribute to these conditions. And so his group, specifically Mari Niemi, who was one of your contemporaries, a PhD student at Sanger, they decided to do a genome-wide association study comparing about 10,000 children with developmental disorders from the Deciphering Developmental Disorder Study to control. So they did a GWAS, and they showed that actually common variants explained about 7% of the variance in risk for these conditions. And specifically through genetic correlation analyses, they showed that it was the common variants associated with a higher risk of schizophrenia that were also associated with risk of these rare neurodevelopmental conditions. And common variants associated with reduced educational attainment and cognitive ability in the general population were also associated with high risk of these rare neurodevelopmental conditions. And so this new paper is basically extending off that work by Mari and Jeff, and it extended on it in several ways. Firstly, we strongly suspected that the common variant component would be greater in patients without a monogenic diagnosis. But actually, in Mari's work, we didn't find that, but we thought that was probably due to low power at the time. And secondly, we thought that there are a lot of new, bigger GWASs since then, and maybe we could learn more about the relationship with other traits by repeating some of those same kinds of genetic correlation analyses. And then finally, we know from GWAS in the field of educational attainment and related traits that a lot of the common variant effects are actually not direct genetic effects on the individual who carries those variants, but they might be mediated through indirect genetic effects or genetic nurture through the family environment. And so we wanted to look at that also in the context of these rare neurodevelopmental conditions. So we brought in new data from the Genomics England study to expand the sample size, as well as a lot more data on parents to allow us to answer those questions.
On that first piece, so let me play it back to make sure that I'm getting everybody else's. There's sort of a relatively common concept outside of neurodevelopmental disorders around this liability threshold model, and you've shown that very clearly that that's the case here. Maybe you could talk a little bit about that and how do you actually establish that that's the case? Because I don't think that was established. It wasn't in the original paper you referenced from 8 years ago.
So in the original paper, which is the Niemi et al., which was published in 2018 in Nature, we just compared polygenic scores that were associated with risk of neurodevelopmental conditions between patients who had a monogenic diagnosis and those who didn't. So I think at the time we focused on the polygenic score for educational attainment because it's the best powered GWAS, and that score explains the most variance in risk out of several polygenic scores that we evaluated. And at the time, we didn't see any significant difference between the diagnosed and the undiagnosed patients. But since then, we increased the sample size, and also we now have a better definition of who's actually diagnosed or not, because we've just done a much better job of finding those monogenic diagnoses over the last few years. So basically, we essentially repeated that analysis, and now we do see a significant difference in polygenic scores. The diagnosed patients tend to have higher polygenic scores for educational attainment and cognitive ability than the undiagnosed patients. Interestingly, in the paper, we also compared both of those groups to controls, and we were quite surprised to see initially that even the patients with a monogenic diagnosis did have lower polygenic scores for educational attainment than controls. But actually, it turned out that that's driven by patients who have an affected parent. And when you remove them, the ones with the de novo diagnosis look the same as controls.
Oh, interesting. Is it fair to say that you could kind of bring the two ends of the spectrum together in some way in the middle? Do we have evidence that there are effectively patients in the middle of the spectrum that have a monogenic hit that in and of itself wouldn't be sufficient to cause neurodevelopmental disorders, but on a particular polygenic background, it effectively tips over the edge into something that would be syndromic? Or is that still a question mark that you're trying to answer?
So I think that's a very likely scenario. And I think Caroline Wright's work in UK Biobank has shown that. So they had this paper, it's Kingdom et al. In Nature Genetics last year. And I'm pretty sure they had this analysis where they evaluated people who had a rare damaging variant in I don't know if it was just developmental disorder genes or loss of function intolerant genes in Biobank, but variants that are strongly associated with risk of neurodevelopmental disorders. And they basically showed that if people had lower polygenic scores for educational attainment, they were more likely to have a diagnosis in their health records. We tried to kind of do a similar analysis using the data from the patient cohorts, a DDD and the Genomics England 100,000 genome study. We basically we wanted to know, we knew that these rare inherited damaging variants were often incompletely penetrant in families. This is based on work from Caitlin Samoka. Often there are affected children who have one of these rare damaging variants that we think is probably diagnostic, and yet it's been inherited from a parent who's apparently unaffected. And so one obvious hypothesis is maybe the polygenic background is protecting that parent. And so we basically compared, this was Emily Wigdor's PhD work, we basically compared the polygenic scores for the affected child to their unaffected parent who'd passed on the variant. And somewhat surprisingly, we actually didn't see any significant differences. So that's kind of contrary to the results in Caroline Wright's work. And that might just be due to power, that the sample size is just not big enough compared to UK Biobank. I think another factor could be the effects of parental assortment, which is something we come to later in the paper. And we show that actually these rare damaging inherited variants tend to be correlated with tend to be negatively correlated with polygenic scores for educational attainment and cognition. And we're pretty sure this is due to parental assortment on traits to do with cognition and education. But because of that correlation, it sort of muddies the waters, and that might explain why we didn't see this evidence, the polygenic background modifying the penetrance of these rare variants within families.
Yeah, can you talk a little bit about that? There's a whole section of the paper on direct versus indirect effects. And this is the first time I've really spent a while learning about them and thinking about them. You were probably wrapping your head around this for months or years before you had the clarity to line it up. So maybe you can help us understand that.
Yes. So this area was not entirely new to me because I've been following the literature on sort of sociogenomics literature on the genetics of educational attainment and cognitive ability for some time. And the real landmark paper in that field was from Augie Kong I think it was about 2018, was it? Where he showed in the decode data that actually quite a lot of the common variant effect that we measure for educational attainment is not mediated through direct genetic effects. So what are direct genetic effects? These are somewhat obviously, the direct effects of genetic variants on an individual who carries those variants. So maybe it's better to explain what they're not. On the other hand, we have what people have called indirect genetic effects, which are the effect of genetic variants, but mediated through relatives who carry the same variants, basically. So you can imagine you inherit variants from your parents that have direct effects on your phenotype, but those same variants are doing something in your parents. Maybe they affect your parents' behavior, and your parents' behavior towards you can also influence your phenotype. Another term for this is genetic nurture. And it turns out that this phenomenon is particularly important in the context of traits to do with cognition and education. But there's some difficulties, which I'll come to. So the way that people often study this and infer that a genetic variant is having a direct effect is they regress a person's phenotype on their genotype, while at the same time controlling for their parents' genotypes. And then when you do this, the effect size you estimate for the index individual's genotype tells you the direct effect of that genetic variant on the individual's phenotype. So this is what we did in the paper. We basically regressed whether or not the child had a neurodevelopmental condition on their own polygenic score plus the polygenic scores of their parents. And we did that for several different polygenic scores. The first one was a polygenic score derived in an independent sample in a GWAS for these rare neurodevelopmental conditions. And for that polygenic score, we did see significant evidence of direct genetic effects. So the child's polygenic score was significantly associated with whether or not they had a neurodevelopmental condition, even once we controlled for the parents' polygenic scores. However, we then looked at the polygenic score for educational attainment, which explained quite a lot more variance than the polygenic score for neurodevelopmental conditions, which is really just because the GWAS is much more powerful. So when we did this, actually the child's polygenic score for educational attainment is no longer associated with whether or not they have a neurodevelopmental condition once you control for the parent scores, whereas the parent scores are highly significant. So that tells us that this polygenic score for educational attainment actually is not having a direct effect on the child's chance of developing these conditions, or if they do, it's very, very small, and we just didn't have power to see it. And that was quite surprising to us because for most phenotypes to do with education and cognition that people have studied, there's usually some evidence of at least some direct genetic effects.
Right.
This is the first that I'm aware of where there was really none, at least for these educational attainment polygenic scores. So what is the interpretation of the parents' polygenic scores being associated with the child's phenotype once you control for the child's score? Well, that finding tells us that genetic variants in the parents that are not transmitted to the child are associating with the child's phenotype. And that can be for several reasons. One is this idea of indirect genetic effects. So maybe the parents' genetics are somehow influencing their parental behaviors, and that's influencing the child's risk. So, for example, maybe the parents' predisposition for educational attainment influences how much they speak to their child and the kind of vocabulary they use, and that affects the child's language development. We know that language delay is a common feature of rare neurodevelopmental conditions, so that's potentially plausible. We have no evidence of that, and we can't test it with the available samples, but that's one hypothesis. Another hypothesis involves a second kind of indirect genetic effect that people often don't really think about, and that is prenatal effects. So the mother's genetics obviously influence various traits in the mum and might influence the prenatal environment that the child is exposed to, and that could potentially affect their risk of neurodevelopmental conditions. So in the paper, we had this section trying to kind of delve into this. We suspected that in particular, so we basically looked at various prenatal factors, including premature birth. I think we looked at preeclampsia. We also looked at smoking, alcohol use and so on. And we looked at genetic correlations between these traits and neurodevelopmental conditions, and also genetic correlations with educational attainment. And we saw that premature birth was very strongly genetically correlated with rare neurodevelopmental conditions, which I think the genetic correlation was like 0.7 or something, which is sort of obvious because we know that premature birth is a risk factor for neurodevelopmental problems. And we also saw that premature birth was pretty significantly negatively genetically correlated with educational attainment, and that mirrors the epidemiological association. So less educated women on average are more likely to have babies prematurely, and there's potentially a bunch of factors contributing to that. Anyway, we suspect that this potential indirect effect we've detected could be mediated by prematurity. So less educated women are more likely to have premature babies, and it's the prematurity that is the risk factor. But of course, they're also passing on genetic variants that predispose the child to more learning difficulties independent of the gestational age. Anyway, we tried to test this directly in a supplementary note by basically controlling for prematurity or excluding children who'd been born prematurely and seeing whether that changed the coefficient on on the parents' polygenic scores. And basically it didn't do so significantly, so we don't really have strong evidence that this is a mediating effect. However, I still think that might just be due to power.
Right.
And I think that it might be part of the explanation, and hopefully in the future we can explore that with larger samples. So then finally, another explanation for why the parents' polygenic scores might be associated with the child's phenotype, even once you control for the child's polygenic score, is parental assortment. So this was a key insight that was given to me by Alex Young, who's one of the real leaders in this field of sociogenomics. We're having this conversation at a conference in Bologna year before last, and he said, "Oh, I think that might be because the polygenic score, the common variants have become correlated with the rare variants that are acting in the same direction on cognition." And just to flesh that out, when people have children with people who have similar traits to them, so that's typically called assortative mating in the literature. We're trying to avoid that term, so we call it parental assortment in the paper. That has several consequences for genetics, one of which is generating correlations between genetic variants that act in the same direction on a trait on different chromosomes. So the common variants on chromosome 1 that are associated with higher educational attainment become correlated with the common variants on chromosome 2. So many people are kind of aware of that. Another thing that happens that's, for the same reason, is that the common variants become correlated with rare variants that act in the same direction on a trait. Whereas in the absence of assortment or population structure, common and rare variants are not correlated with each other. And so Alex said, and has kind of showed through his work, that the common variants in the parents that are not transmitted to the child can become correlated with rare variants that are transmitted. And we show empirically in our paper that indeed the burden of rare damaging variants that are associated with higher risk of neurodevelopmental conditions and lower cognitive ability, they tend to be correlated with lower polygenic scores for educational attainment that are based on the common variants. So we show that this correlation exists. We tried to test whether, in fact, if you control for these rare variant scores, does that affect the coefficient on the parent's polygenic scores, to see whether these transmitted rare variants are actually mediating the effect of the common variants in the parents. And we didn't find that, but we're pretty sure that's just because the polygenic scores and the rare variant burden scores we're using are only capturing quite a small fraction of the overall variance due to common and rare variants. And so if we had perfect scores, I think we could draw a different conclusion. But anyway, that insight basically made us realize that all these results we've generated to do with common variants could potentially be confounded to some extent by their correlation with rare variants.
Right.
And so that's really future work, trying to disentangle that. And it's going to need very, very large samples, including of within family studies, basically, including multiple generations, not just the parents and the child, but also the grandparents, which allow one to disentangle this confounding due to parental assortment from true indirect genetic effects or genetic nurture effects. And Alex is really leading theoretical work in that space.
Is there anywhere you can do that work besides Finland or Iceland, places where they have very significant, long-standing, generation scale health programs?
Well, I mean, one of the challenges is that when people collect patients with rare conditions, they might collect their parents, but they almost never collect their grandparents or their aunts and uncles, which is what we need to apply these 3-generation models. However, we may be able to get to this, at least in the context of cognitive traits measured in the general population or educational attainment, using birth cohort studies. Particularly, there's this big study, MOBA, in Norway, which has 100,000 babies. And at the parental generation, there's lots of siblings. And Alex has shown that once you have siblings, you can impute the parental genotypes. You don't actually have to measure them directly. he has a Nature Genetics paper about this. And if you then can control for those imputed ground parental genotypes, then it allows you to clarify your inferences to some extent and remove these confounders. So that's a direction we are heading in.
Interesting. And we've gone many layers down the tree, but if we kind of come back up to the very top, I have two other questions on this. One is just around if we think about the families and the parents at the center of all this. You did one thing in this paper that was really interesting, which was you put together actually a lay summary and FAQ. I'd love if you could talk a little bit about that, because it goes without saying, this is an extremely sensitive topic that can be, this parents really want to understand what might be happening in their case. And also whenever educational attainment and cognition gets involved, people can tend to take results like this and try to make them mean things that they don't really mean. So I would love if you could just talk a little bit about the process, but putting together a lay summary and how you went about that.
Sure. So yes, we realized fairly early on that it would be important for this paper. And we reached out to the participant panel at Genomics England that's associated with the 100,000 Genomes Project, and asked if there would be people there who'd be willing to help us draft some sort of FAQ document, which has become commonplace in the sociogenomics world. And we were very lucky that Gillian Hastings-Ward, who at the time was the head of that participant panel, was willing to help us, and she later wrote in some other people who also contributed. And initially we kind of wrote a draft and sent it to her, and she sort of tore it to shreds and told us how a lot of the language was problematic, or it wasn't answering the questions that families would want to know the answers to, and it was all too complicated and so on. So we had to go through quite a few iterations, and her input was really valuable. And I think by the end we ended up with something that they were, I think, fairly happy with, and we were happy with. And we definitely learned things through doing that. So one of the things we did is, so in addition to this FAQ, which is quite long, it's sort of 20 pages long, there's also just a lay summary, which is about 1 page. But even that, they said this is kind of too challenging for a lot of people, particularly people with mild learning difficulty who might be interested in the results. So can you write an easy read version, which is this format that the NHS often uses? And so we wrote an even more lay version to try and convey the motivation for the study and what we found and what the results mean and don't mean.
The related question that was, this has got me thinking to what kind of effect this might have when you work all of this out, what's actually going on that's driving this, the impact that it has on actually how you do clinical testing and help people navigate what is actually the cause here. Because in the monogenic world of this, the story is quite clear. You have a rare, very damaging variant that's messed up this particular gene, and that has caused this particular problem, and that may even lead to a treatment, right? If we can, deliver a healthy copy of this gene and get the levels up to where they will be, then maybe there's a way to treat it. In this polygenic, monogenic combination or extreme polygenic world, it starts to become a lot more difficult to return results, explain what's going on. I know we're not there yet, but I wondered if you thought a little bit about what implication this might have on returning results to families.
Yes, indeed. It is much more complicated, and I think there's a lot of work to do. I mean, I definitely don't think we're ready to be doing that, because firstly, we're not perfect at interpreting the rare coding variants we find, right. And there's probably a lot of people with VUSs that are passing our filtering, but where we just don't yet have enough information to decide that they're definitely pathogenic. But we probably will be able to do that increasingly in the next few years, particularly with saturation genome editing assays and so on. And then there's the whole non-coding genome, which we've barely touched. So I think, ruling out that there's a very large effect non-coding rare variant causing a patient's condition is going to be pretty difficult. But yes, I think it's quite plausible that, in 10 years or more, we might be wanting to return information about polygenic risk to families. And one thing that I think might happen sooner rather than later, and I'm not sure if this necessarily will ever need to be conveyed to families, but we might want to start using polygenic risk to kind of help us find the right large effect rare variants in a patient's genome. So, it might be that a patient comes in who's got, learning difficulty and heart defect. And so you go looking for some rare variant that causes both of those, and you find one that causes the heart defect, but it doesn't explain the learning difficulty. Well, if they had a really low polygenic score for educational attainment, then, maybe that is actually the explanation. And so we want to kind of start exploring that at the research level. So I can imagine, and if we find evidence that that's the case, then I can imagine that clinical geneticists might, in 10 years more, start incorporating that information into trying to identify large effect rare variants. But yeah, I think there's a lot of work to do to A, figure out whether this is diagnostically useful, and B, figure out how to communicate it to people.
Yeah. I mean, it is. You made a great step in the right direction, though. I want to talk a little bit about the other branch of your work, which is consanguinity and diseases that tend to be more common or more impactful in those populations, and in particular, some of the work you do with East London Genes and Health. How did you get interested in that area of research? And maybe you could talk a little bit about some of the projects you're involved with.
Well, it was really two strands in parallel, I guess. When I was finishing my PhD, I became aware of the East London Genes and Health project. Well, as it was then called, it's now just called Genes and Health, which is a study that aims to improve the health of British South Asian people, but specifically British Pakistanis and Bangladeshis, through doing genomic research. And it was known that this population had relatively high levels of consanguinity, and as a consequence had more rare gene knockouts, so homozygous rare variants, including loss of function variants, and those were particularly interesting for drug target discovery. And so when I came to Sanger, initially I started working with Richard Durbin on this project, but then there weren't really a lot of data from it yet, basically. So at that time, I decided to kind of sidestep into working on recessive disorders in the Deciphering Developmental Disorder study with Jeff Barrett and Matt Hurls. And so we did this project basically trying to quantify the contribution of recessive variants to rare developmental disorders in the DDD study. And I've done follow-up work on that, which was led by my postdoc, Kartik Chundru, and we just published that in Nature Genetics last year as well. But then the Genes and Health study was kind of rumbling along in the background and accumulating more samples. Now they've got 55,000 people. We lucky that we were able to establish this industry consortium with a bunch of pharma companies who put in money. So now the whole cohort's being exome sequenced. So now it's really become a really fantastic resource for doing genetic research in a non-European population. And so we've been doing a variety of projects there, partly to do with population structure and partly more medical genetics. And a lot of it's kind of focused on the autozygosity and looking at effects of autozygosity on complex disease, and then following that, looking for specific variants in genes with a recessive effect on complex diseases and traits.
And you look across the two strands do share that common origin in developmental disorders. When you look at what you and the team are going to focus on for the next couple of years, what are you most excited about?
So I'm actually most excited about extending the work from this paper I just talked about on the common variants in neurodevelopmental conditions. I think that paper, like many papers, kind of prompts more questions than it answers, really. So I'm most excited about trying to get to the bottom of, why are these non-transmitted variants in parents associated with the child's risk? And that's useful because if we can show that there's a kind of causal effect due to genetic nurture as opposed to this sort of boring consequence of this sort of artifact of parental assortment, then maybe it suggests that some kind of interventions could help improve outcomes for children with neurodevelopmental conditions, at least some of them. So I think I'd really like to get to the bottom of that question.
Along this similar topic, there was a recent preprint from your group as well, looking at the differential effects of common and rare genetic variants on cognitive performance in a couple different birth cohorts. I wonder if you could talk a little bit about that as well, because I thought it was interesting.
Sure. So this was motivated by this observation I referred to earlier, that quite often children with rare neurodevelopmental conditions inherit a rare damaging variant from their parent, that looks like it might be the cause of their condition, but the parent is unaffected. So clearly this rare variant is incompletely penetrant. And, one potential explanation for that incomplete penetrance is modification by polygenic background, which we talked about earlier. A second hypothesis, which is not mutually exclusive, is that these rare variants have larger effects on cognitive ability in early childhood that then attenuate with age. And so sometimes people kind of grow out of the effect of the variant. And so, which is why the parent appears to be unaffected. And we'd heard anecdotally from clinical geneticists that sometimes in families like this, when you start asking questions, it turns out that the parent did actually have learning difficulties when they were a child and they're in the special school, but they've kind of caught up cognitively and in the normal range, basically. And so we wanted to test this we being Matt Holes and I, and so we decided to go and sequence first cohorts where they had measured cognitive ability and related traits repeatedly throughout childhood and adolescence all the way through to adulthood, so that we could look at the effect of genetic variants on those traits at different time points. And so Daniel Milowski, who's a PhD student in my group, he was the main analyst for this, and basically we've just released a preprint with the results, and he shows that that actually this hypothesis seems to be correct, that these rare damaging variants, they are associated with reduced cognitive ability, but particularly in early childhood. The measure we have in our SPAC is at age 4, and that then attenuates as they get older. In contrast, common variant-based polygenic scores for educational attainment seem to explain more variance in cognitive ability as people get older. And we use quantile regression to basically look at whether these common and rare variants differentially impact different parts of the phenotype distribution. And we basically show that the increasing effects of the common variants are really stronger in people at the upper end of the phenotype distribution, so with higher IQ, whereas the attenuating effect of the rare variants is stronger at the lower end. So that finding is really kind of consistent with the observations in cohorts of children with neurodevelopmental conditions, that these rare damaging variants might appear to be incompletely penetrant within families. And in some cases, it might be because of this age effect, that people might have difficulties when they're younger that then attenuate. And so what we'd really like to do is understand why the effects of these variants are attenuating. Is it because children have difficulties early on, kind of targeted for more help at school or at home, and that helps them to kind of recover from the effect of those variants? Or is it something kind of biological? Yeah. And if we could answer that, because if it is due to nurturing effects, then that suggests that there are interventions that might help improve outcomes for people with early neurodevelopmental difficulties, at least in some cases.
Yeah, that's super interesting. It's almost like the inverse of the penetrance problem that you normally see in biobanks, which is the penetrance increases with age for something like a C9orf72 variant in ALS or FTD or ApoE4.
Right. Yes,
but it's almost the inverse of that.
But you're right. Yeah.
Yeah, that's very, very creative, interesting work, and always cool to see some of the anecdotes that come from clinicians looking at these things for many decades actually being tested and borne out by data.
Well, sort of tested indirectly. What we'd really like to do is to have cohorts of people with neurodevelopmental conditions, but have much better phenotype data on their parents and relatives. So we could kind of test this more directly in a quantitative way. But, it's a sensitive topic. And so it's quite hard to convince people that this is a good idea. But anyway, ongoing.
It seems like a lot of this work is very constrained by access to data sets, right? You're pushing the data sets that you have to their absolute limit.
That's definitely true.
Yeah, interesting. Yeah. When you were talking about direct and indirect genetic effects, I was searching for a metaphor in my mind, but realized I had one staring right at me, which was actually around your background, how you got into genetics. You have both your parents are geneticists. You have a grandfather who's a geneticist as well. That is sort of a toy example, right? Just genetics in the family, nature or nurture. Talk a little bit about that. That's
genetics is hereditary. Yes.
Yeah.
Yes, it's quite unusual. I mean, I was extremely lucky from my perspective. I was born to two geneticists, two human geneticists, and it's actually my father's father was also a kind of an early geneticist, really trained as a botanist, but he did what was genetics in those days, which was mostly kind of staring down microscopes at chromosomes. So yes, obviously from an early age, I was exposed to what was going on in the field, and also until kind of the end of first year university, I kept an open mind, and I thought I'd go into some branch of science, but not necessarily genetics. Actually, that's what really
couldn't help yourself.
Really floated my boat. So yes, I have ended up following in the family trade.
You've published some papers together, right?
A little bit, yeah. My dad's contributed data to one of my PhD papers. I've contributed in some minor way, I can't even remember, to some work of my mum's. Yeah, we have a little bit.
Do you, when you go home for, Australia's a long way to go home for Christmas, but when you do go home for Christmas, do you have a no genetics or all genetics all the time?
Well, it depends whether my sister's there or not, because she's not a geneticist. If she's not there, then there's a lot of genetics talk.
That's great. I'd love to hear the platypus story, actually. As we wrap up here, your grandfather, I think, wrote a textbook that had the karyotype of the platypus wrong, but you gave him a pass because it was in the 1960s, right? Tell that story.
Well, look, there's not much more to tell, really, except that one of my PhD was on extremely disparate topics, one of which was the population genetics of the platypus. And the platypus, we now know, has this very funky sex chromosome system of five different X chromosomes and five different Y chromosomes that all line up in this long chain at meiosis, so that all the X's can go one way and all the Y's can go the other way, and they give rise to female or male. The sperm go on to fertilize female or male platypuses. Anyway, so we know that now. I discovered during my PhD that my grandfather had done a bunch of work on his name was Peter Martin, by the way, Peter Gordon Martin. And
you got to look up his work.
Done all this work on the chromosomes of Australian mammals, and which, was just kind of classical cytogenetics in those days. But anyway, this book, that it had the karyotype of the platypus wrong, which is not really surprising given how complicated it turned out to be. But nonetheless, I dedicated that particular chapter to him.
Oh, I love that. Well, Hilary, thank you. I appreciate you taking the time to be with us today. Is there anything you're looking for? Collaborators? Are you hiring postdocs, PhD students, or what do you want people to? How should people reach you if they're interested in collaborating?
Well, they can obviously email me. I don't have any open positions at the moment, but I may well in the next few years, and I'm definitely always happy to hear from keen people who and can potentially get money to hire them if they seem like a really great fit. I'm definitely interested in hearing from people with cohorts that have lots of relatives, either three generations of relatives, or cases where you've got parents, children, and siblings of those parents, and who have phenotypes to do with neurodevelopment and cognition. I'm aware of some of those cohorts and we're already working with them, but if you have any that you think that I might not know about and where the data are open or you're keen to collaborate, do get in touch.
I wondered if there might be a way to link together UK Biobank and the newborns program. And I've heard rumblings that there might be a Generation 2 UK Biobank. It seems like you might be able to do this in the UK with the new newborns program, a few others, by making linkages between the two or three different programs. Do you think that's possible?
Yeah, well, yes, those discussions are happening. There's definitely quite a lot of enthusiasm for extending UK Biobank to another generation. That's kind of a major undertaking that would require a lot of funding. So another option, as you say, is linking to other cohorts that are already being collected. I know we're having discussions about, would it be possible to link UK Biobank and our future health? Right. And I think those discussions are in the early stages, and probably many people are having that conversation in parallel. I don't know if there's been discussion about linking the newborns program to other cohorts, but obviously that would be fantastic if it could be done. There's various kind of governance and logistical challenges, but I think that's a conversation that we should definitely have in the coming years.
Yeah, definitely. Well, Hilary, thank you. I appreciate it. And thanks everyone for listening. We'll see you next time.
Thanks very much, Patrick.
That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Cynogenetics on Twitter, LinkedIn, or visit cynogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible, in particular Amy Cousins and Sonia Shah who produced the show, and James Pierce from Selected Frequencies who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.